Otonude. Do you hear me? Tell me what I discussed in my last class. No, did I discuss the structure of nerve? Okay, that means neuron, ependymal cells, neuroglia, and neurosecretory cells. Did I discuss their function or structure of myelinated and non-myelinated neuron? No. Discussed. That means now did I discuss how impulse is propagated through a neuron? Oh, discuss it. Did I discuss synapse? That means I discussed up to the difference between myelinated and non-myelinated neuron, and I discussed how impulse is propagated through it. Whenever it is a polarized state, whenever it is depolarized, and whenever it is repolarized again, right? That means?
Yes, sir.
Okay. So one thing is that the synapse. The junction between two neurons called synapse, and synapse is of following types. Exoexonic, exosomatic, this is the common type. In case of exoexonic type, you see generally this is the exodendritic synapse. And here, this is the axon part, this is called presynaptic membrane. Postsynaptic membrane is the axon part. Here you will get they are called synaptic vesicle. Filled with neurotransmitter. This swollen part is called synaptic nerve. In the postsynaptic membrane, you will get the receptor. Receptor there is actually space, fluid-filled space between two neurons. This fluid-filled space is called synaptic cleft. Different ions are found to be present here. So this is the structure of synapse. Presynaptic membrane is the axon. If it is the exodendritic synapse, in the exodendritic synapse, the presynaptic membrane is the membrane of the axon terminal, and postsynaptic membrane is the membrane of the dendron. There is a space between these two, which is called synaptic cleft, and which is fluid filled. Different ions are, the fluid is called extracellular fluid, and different ions are found to be present there. Clear? Different ions are found to be present there. Now what next? After that you see, whenever normally the membrane of the presynaptic membrane normally these synaptic vesicles are stimulated by calcium ion only. But calcium channels are found to be closed when in resting stage. So whenever the stimulus comes, the permeability of the presynaptic membrane changes. Calcium ion channels open. So from here calcium ion enters inside, stimulate this vesicle, and vesicles now comes in contact with the presynaptic membrane and secretion starts. That means basically the neurotransmitter, mainly acetylcholine, is present, so secretion starts. Now this acetylcholine will then bind to the receptor. As soon as acetylcholine binds to receptor, permeability of the postsynaptic membrane changes, so that from the extracellular fluid, sodium ion enters, and due to the regular entry of sodium ion into the presynaptic area, postsynaptic area, a gradient is developed which is called action potential. So once action potential is developed, means impulse is carried to the dendron site. This is how impulse is propagated through the synapse. Such a type of synapse, is called chemical synapse. This type one is called chemical synapse. Chemical synapse is a synapse that is formed where you will get in case of chemical synapse, there is actually in between the presynaptic membrane and postsynaptic membrane a fluid filled space is present, which is not found in the other type of synapse called the mechanical synapse, actually electrical synapse. Remember this thing, synapse is of two types. I should write here. Wait. Electrical synapse, chemical synapse. Here, pre and postsynaptic nodes are in In such synapse, neuron in such synapse, presynaptic neuron and postsynaptic neuron are in very close proximity. Such synaptic cleft is not developed in this case. Synaptic cleft is not developed in this case. What happens here? Presynaptic membrane and postsynaptic neurons are in very close proximity, and impulse is propagated quickly through such synapse. But such type of synapse is very rare in human being. Electrical synapse is very rare. In our nervous system. Electrical synapse are very rare in our nervous system, but the chemical synapse, in case of chemical synapse, a fluid filled space is present between presynaptic membrane and or presynaptic neuron and postsynaptic neuron, which is called synaptic cleft. Is it clear to you? Is it clear? Electrical synapse and chemical synapse?
Sir, can you tell the part of chemical synapse again?
Write down. Is chemical synapse, as I am writing here. A fluid filled space is present between presynaptic neuron Look here. In chemical synapse, a fluid filled space is present between presynaptic neuron and postsynaptic neuron. And you know here, impulse is propagated with the help of a chemical called neurotransmitter. Chemicals called neurotransmitters help in the propagation of Is it clear? What is it then? In chemical synapse, a fluid filled space is present between presynaptic neuron and postsynaptic neuron. As you know, it is called synaptic cleft. Chemicals like neurotransmitters help in the propagation of nerve impulse from presynaptic neuron to postsynaptic neuron. Clear?
Yes, sir.
Is it clear now? Any doubt you can tell me.
Yes, sir. Clear, sir.
So this is all about the nerve tissue. Already I discussed, I think, about the types of neuroglia and role of ependymal cell and also that of the neurosecretory cells. So I'm going to rub it out now. Can I rub it out?
স্যার একটু দাঁড়াও
আচ্ছা আচ্ছা ঠিক আছে করো না।
ডান স্যার স্যার ডান
Okay. Now the animal tissue completed. We have to proceed towards
স্যার
the
নিউরোগ্লিয়ার জায়গাটা স্যার
হ্যাঁ কোনটা?
স্যার টাইপস অফ নিউরোগ্লিয়া স্যার এই জায়গাটা করনি তো
ও
স্যার শুধু নিউরন করিয়েছ?
আচ্ছা আচ্ছা আচ্ছা।
স্যার নিউরোগ্লিয়া, তারপর অ্যাপেন্ডাইমাল সেলস এগুলো করানো।
Neuroglia are supportive in function. They do not take part in nerve impulse propagation. Supportive in function and do not take part in nerve impulse propagation. These are two types. Macroglia. Larger neuroglia. Astrocyte. Mechanical support. Oligodendrocytes. Myelin sheath. Microglia, smaller neuroglia, function as small macrophages. That means they can engulf several bacteria, microorganisms found in the system. So this is what about the neuroglia, two types. They are basically supportive in function and do not take part in nerve impulse propagation. So 50% of the space of the brain is actually occupied by the neuroglia, and when a neuron dies, its space is also occupied by the neuroglia. They are to be supportive in function. Two types of neuroglia found: macro mean larger, micro mean smaller. Macro are two types. One is star-shaped astrocytes. is called, actually this is the astrocyte. And oligodendroglia is also a larger cell, like if you like this. So this is what about the two types of nerve cells. Is it clear? And those who are
Yes, sir.
macrophages, these are macrophages. Microglial cell. Here they can ingest bacteria. Suppose they are ingesting bacteria here. So this is what about the neuroglia in short. Next comes to the, after neuroglia, then the ependymal cells. Can I rub it out?
Yes, sir.
Sir, please wait.
Huh?
Sir, wait.
Completed?
No, sir, wait.
No, sir.
Okay. Neuroglia are supportive in function. They provide support to the nerve cells. Remember these things. And they are neurons and nerve fibers. Supportive in function, বুঝে তো জিনিসটা? Neuroglia are supportive in function means they provide support to neurons or the nerve cells or nerve fibers of brain and spinal cord. Their supportive in function means they provide support to nerve cells, nerve fibers present in both brain and spinal cord. They do not take part in nerve impulse propagation. There are types given here.
Sir, done.
Okay, thank you. Ependymal cell. They act as these cells are one cell thick epithelial cells. Now these cells behave like epithelial cell. Clear? And found lining the cavity of brain and central canal. Epithelial layer of one cell thick. Like the cells are arranged as an epithelial layer of one cell thick. They are found, it is found to line the ventricles of brain. It lies the ventricles of brain and central canal of spinal cord. Ventricles of brain and central canal of spinal cord. At their free surface, microvilli are developed. Their free surface possesses microvilli. Microvilli which help in the absorption of cerebrospinal fluid. There are microvilli which help in the absorption of cerebrospinal fluid. You know, in the ventricles of brain and in the spinal cord, you will see it appears like this. So look here. Up to this part, you can call it brain. From here, spinal cord. Is it clear now?
স্যার ওই সাইডটা দেখা যায় না একটু
দেখাচ্ছে এবার?
হ্যাঁ স্যার ঠিক আছে
So this is the role of it. Now the next one is neurosecretory cells. Specialized nerve cells secreting neurohormones. Example, neurosecretory cells of secretes neurohormones like
স্যার ওইটা দেখা যায় না স্যার
দেখবি oxytocin ADH. Neurosecretory cells, the last type. These are specialized nerve cells secreting neurohormones. These are specialized nerve cells secreting neurohormones. Example, neurosecretory cells of hypothalamus. Neurosecretory cells of hypothalamus secrete neurohormones like oxytocin ADH. Clear? Is it clear? Clear?
Yes, sir.
So this is all about the nerve tissue. So animal tissue is completed in this way. Okay, it's a very simple thing I'm going to ask you. Can I rub it out now?
No, sir. No, sir.
Huh?
No, sir.
Okay.
স্যার
Yeah, yes.
স্যার ডায়াগ্রামে স্পাইনাল কর্ডের উপরে কি লেখা?
Oh, brain.
আচ্ছা ঠিক আছে।
Done, sir.
Clear? Now, a very simple idea I'm giving you, nerve. Whenever the term nerve is used, whatever consists of it consists of several bundle of nerve fibers. And each bundle is called fasciculum. Each bundle is called fasciculum. Remember this thing. It is surrounded by a Perineurium. If I am going to draw a diagram, then it will be easier for you. Look one thing. Look. This is the epineurium.
Sir, sir,
হ্যাঁ
each bundle is called কি লিখেছো এটা?
fasciculum, Fesciculum.
Okay, sir.
This is the epineurium. Perineurium. Androneurium. Okay. Now look at clear. Here you will get some blood vessel. This is what nerve. These are the several bundles of nerve fibers. Each bundle is called fasciculum and it is protected by a membrane called perineurium. This is also perineurium, this is also perineurium. And each perineurium is again made of flattened cells surrounded by androneurium. So this is the androneurium. This part is the androneurium. And inside which you will get the axon and surrounding which you will get the myelin sheath. So there's a whole, the structure of nerve. Is it clear? So this is what about the nerve, and I think it's clear that what are nerves? Some are sensory nerves, some are the motor nerves, some are mixed nerves. So nerve may be or you can write afferent nerve. Afferent and mixed. Okay, just a minute, please. Just 1 minute coming. Made of motor fibers. Made of both sensory and motor fibers. Made of afferent neuron made of sensory fibers, efferent made of motor fibers, and mixed made of both sensory and motor fibers. Clear? So this is what about the nerve. Afferent neuron sensory fibers carrying information from receptor to CNS. So afferent neuron carry impulse from receptor to CNS. Carry impulse from CNS to receptor. In both directions. Here impulse, that means here the mixed nerve, as it is made of both sensory and motor fiber, the sensory fiber carries impulse from CNS to receptor to CNS, and motor fibers impulse carry impulse from CNS to effector. Clear? Is it clear to you? Afferent, impulse from receptor to CNS, efferent, impulse from CNS to receptor, effector, and mixed, as it is made of both sensory and motor fibers, it carries impulse. The sensory fibers of mixed nerve carries impulse from receptor to CNS, and motor fibers of the mixed nerve carry impulse from CNS to effector. Effector means muscles, effector means gland. Clear?
Yes, sir.
Okay. So this is what about the nerve already I discussed. Then here nothing more important. Already I discussed the myelinated and non-myelinated neuron also. And so this is all about this. Now sometimes neurons maybe you can go through it. Depending on the polarity also you can classify neuron like non-polar, unipolar, bipolar, multipolar. So this is what about the nerve tissue. You go through it. Is home task unipolar neuron, bipolar, non-polar, without polarity, non-polar, unipolar, bipolar, multipolar neuron. It's your task, you need to go through it. It's your task. What do you mean by non-polar, unipolar, bipolar, and multipolar neuron? On the basis of the structure. This classification is based on the classification of neuron based on structure. Is a classification of neuron based on structure. The types are non-polar, unipolar, bipolar, and multipolar neuron. And you yourself to find it out. This is your task. Clear? Is it clear? You yourself find out this. So this is all about the animal tissue. Now, a part of nervous system, a part of the, blood, body fluid circulation, a part of movement and locomotion also completed in this way. Now we have to proceed towards the unit 4. Can I rub it out now? Huh? Oh, that means actually obviously only 3 types of muscles are there: skeletal muscle, cardiac muscle, and smooth muscle. I discussed the skeletal muscle only. You are saying like this. They are also very simple. Regarding the smooth muscle, they are spindle shaped cells. They are spindle shaped. They are the muscle fibers. No striation is found actually. They are the smooth muscle fibers. Sarcoplasm. So in case of smooth muscle fibers, you remember this thing basically. This plasma membrane, they are uninucleate. The cytoplasm of the muscle cell is called sarcoplasm. Basic function of smooth muscle is to help in the movement of visceral organs. The main function of the smooth muscle is to help in the movement of visceral organs. Mainly it helps in peristaltic movement. Helps in peristaltic movement. Smooth muscles help in peristaltic movement. Clear? Okay. In case of cardiac muscle, so it is found in the visceral organs mainly. Cardiac muscles, I think I discussed that why cardiac muscle never get fatigued. And in the cardiac muscle, you will get some intercalated disc to join different cardiac muscles together. Suppose in this way, they are found to be connected, and here you will get some disc-like structure so that it is appear like that of the single fiber. Cardiac muscles are joined by intercalated discs so that they behave like a single muscle fiber which helps in propagation of nerve impulse. This is what about the cardiac. Each fiber is uninucleate, but they have dark intercalated discs at regular intervals, and so that they can behave as a single muscle. And it is required for actually conduction of the impulse to the cardiac muscle fiber. If they are to behave as a single unit, then only impulse can be propagated smoothly and uniformly through the cardiac muscle, which is very much required. That's why intercalated disc developed in cardiac muscle. Clear? Is it clear to you?
Yes, sir.
Each fiber is uninucleate, but they are joined by dark intercalated discs so that they can help with uniform conduction of impulse, which is very much required for the functioning of the heart. Each fiber is found to be uninucleate. And why cardiac muscle never get fatigued, already I discussed. Can you remember? Cardiac muscle never get fatigued. Why? This is because in cardiac muscle amount of myoglobin is more, its refractory period is long, and lactic acid cannot be accumulated in the cardiac muscle due to the presence of LDH1 type of lactate dehydrogenase enzyme, which is inhibited by pyruvate. Clear? I discussed it, I think, na? Why cardiac muscle never get fatigue? I discussed it earlier. Amount of myoglobin is more, the refractory period of cardiac muscle is long, and lactic acid cannot be accumulated in the cardiac muscle due to the presence of LDH1 type of enzyme, which is inhibited by pyruvic acid. That's why lactic acid cannot be found. And as lactic acid cannot be found, so it cannot be fatigued. Clear?
Yes, sir.
Okay. So this is in short about all types of muscles. Now, what I am going to discuss, you have to issue home task, differentiate between, write down, differentiate between skeletal muscle, smooth muscle, and cardiac muscle. Clear? Differentiate between cardiac muscle, skeletal muscle, and smooth muscle.
sir.
Okay, বল।
Sir, কয়টা points দিতে হবে?
হ্যাঁ 4 points, not more than 4 points.
Sir, এমনিতে mark এটার কত marks থাকবে?
কোনটা? Difference?
ওই তো,
2-3 marks.
আচ্ছা।
Actually 4 marks, what I am going to give you for 4 points, because it will be helpful in the answers, that's why.
Okay, sir.
So this is all about the animal tissue, completed animal tissue now. Just wait a minute, please. Just coming. Next part, I am to start with the unit 4. Can you tell me which chapters are deleted in unit 4? Anyone, please? Can you tell me which chapters are deleted? Abrojit, Ishita, Tanushree. Can anyone tell me, please? Do you hear me? Annesha, do you hear me?
Yes, sir.
Now you tell me, in the unit 4, I think 2 chapters are deleted, are in the deleted series. What are these 2 chapters?
Sir, I don't know.
Huh?
Sir, I don't know.
Oh, you don't know. So we have to start with the photosynthesis. Actually, I am going to leave you today. I have some other function, actually I have some other tasks. So I am going to leave you today. For my next class, I am to start the unit 4. Okay?
Sir,
Yes.
sir, you told that 2 chapters are deleted, but here 5 chapters are there.
Which one?
Which chapter? Sir, unit 4.
You do one thing, Monishika. You just can you send it to my WhatsApp?
Okay, sir, I am sending right now.
That's fine. That's fine. So in my next class, I am to discuss this unit 4. I am to start unit 4 in my next class. Okay?
Okay, sir.
Okay. Thank you.